Okay, now we're set. Getting a late start because of the class ahead of us. How's everybody doing today? All right, so I had one last tiny thing I wanted to say about nucleotide metabolism, and then we'll sort of change directions in the course and start talking about molecular processes, DNA replication, transcription, translation, and a variety of related topics, gene expression, sensory systems, immune system, and so forth. But before I do that, I had one last thing I wanted to mention, and this was this enzyme, HGPRT, that was mentioned in salvage synthesis. It's actually involved in both catabolism and the salvage and synthesis of purine nucleotides. This guy here, HGPRT, which stands for hypoxanthine guanine phosphoribosyl transferase, I believe. Okay? But you'll probably know it as HGPRT, which is better than Cletus or something like that, is an enzyme that catalyzes a relatively obscure reaction that involves the salvage of guanine nucleotides. So when we look at the catabolism of guanine nucleotides, we see that adenine or guanine can be combined with PRPP to make adenylate, which is a nucleoside, right? So it's got a sugar and a base. I can't say the word. As well as guanylate. These are both nucleosides. It turns out hypoxanthine that you saw earlier on that breakdown product in gout can also be salvaged and put onto PRPP. And if you put hypoxanthine onto PRPP, just like you do these guys up here, what you make is inosinate, and of course that's the nucleoside that becomes IMP. Okay? And actually, I'm sorry, this has the, that is IMP. So inosinate's the same thing as IMP. At that point, you have a nucleotide that is the branch point then for the synthesis of AMP and GMP. So what this enzyme HGPRT does is it catalyzes the formation of this reaction, that is the formation of IMP from hypoxanthine, which is technically a breakdown product in purine metabolism. So you see something being broken part of the way down and then salvaged and made back into purine nucleotides. It would seem to be a relatively minor enzyme, but deficiency of this enzyme is linked to Lesch-Nyhan syndrome, which is a very severe neurological disorder and causes immense problems. And I'll leave it at that. But HGPRT is a salvage enzyme that's salvaging a catabolic byproduct of purine metabolism. And it's important in providing a pretty good source of IMP for the synthesis of AMP and GMP. Okay? So this is the first salvage synthesis that you've seen. I'm not going to talk about pyrimidine salvage at all. But this salvage is important because imagine, well, you might say, well, why would you be doing this? All right? If you think about what you get in your diet, all right, you think, well, I get all this protein, I get all this carbohydrate. You're also getting nucleic acids in there. And your body is breaking those nucleic acids down in the digestive system. You have enzymes that not only break down proteins, but they also break down the other components, the carbohydrates, and nucleic acids. Those nucleic acids have useful material in them, like purine nucleotides, that can be salvaged, taken to cells, and then cells can start with those materials and build full-blown nucleotides without having to start from scratch. So salvage synthesis is an important component of nucleotide metabolism. Okay, that's kind of quick. That makes sense? Okay. All this enzyme does, it catalyzes this reaction right here. That's all it does. Okay. Well, believe it or not, after a long haul, we have now finished metabolism. We will not talk any more about metabolism. And in fact, we turn our attention now to processes of DNA replication, recombination, and repair. Most of what I will have to say here will relate either directly or indirectly to replication, I will talk about repair only in a fairly brief sense. Okay? Now, when we talk about DNA, everybody's had some of the basics of DNA. We talked about them a little bit last term, and I'm going to show you some of the things in a little bit more depth this term. The structure of DNA, of course, was one of the most important scientific discoveries of the 20th century, if not the most important. You could put Einstein's theory of relativity and the structure of DNA probably on equal footing. And depending on whether you're a biologist or a physicist, you would argue about which one was more important. Probably the one that has impacted our daily lives the most has been the knowledge of the structure of DNA. Okay? The biotechnology revolution that's out there, the molecular medical approaches that are out there, the design of new drugs, all of these come from, ultimately, our knowledge of the structure of DNA. Okay? So, I want to say a little bit about that, about both the structure and sort of just some replication considerations, and then get into some details specific to the structure. When we look at the replication of DNA, we see a figure like this that is pretty common textbook material, and we take something for granted here. Okay? Well, I know how DNA replicates. You know how DNA replicates. Each strand is copied from the other one. Okay? That information is built into the DNA molecule. All right? Before people started studying replication, they didn't know this. Okay? People had to do research to show this. There's two scientists who were credited with discovering how replication proceeded. That was probably as important as the structure of DNA, but a lot of students don't know them. Okay? Their names were Messelson and Stahl. Frank Stahl was actually later a professor at the University of Oregon for many years. He only retired from the University of Oregon a few years ago. Very important experiment that they did that showed replication occurred like this. And you'd say, well, what other way could it occur? Well, imagine that I had a single parental molecule up here that's in blue, and in copying it, it would be like taking it to a photocopier. I make another copy over here that has two strands red. Okay? That's a conceivable way of replicating. It doesn't tell us how we got from one to the other, but that's a conceivable model of replication. That model of replication is called conservative replication. Okay? It's conservative in the sense that we don't alter the original. All right? Well, it turns out that DNA does not replicate that way. And what Messelson and Stahl showed was that, in fact, that DNA replicates by what we refer to as a semi-conservative model. Okay? It's semi-conservative model. Each daughter strand has one original strand and one newly synthesized strand. Semi-conservative replication is the way that our cells replicate. It's the way that probably 99.9999% of cells in the world replicate. As part of my PhD thesis, I was actually able to show a virus that did not use semi-conservative replication. It's an oddball, to be sure, but it's the only one I'm aware of that does not use a semi-conservative model of replication. So semi-conservative model of replication is everywhere, and it makes very good sense. The reason it makes very good sense is there's enough information in one strand to duplicate the other. Well, that sprang ultimately from the knowledge of the, I won't talk about that. Sprang ultimately from the knowledge of the structure of DNA that was determined by Watson, Crick, and I also like to give credit to Rosalind Franklin. Okay? So when we talk about the structure of DNA, James Watson, Francis Crick published a landmark one-page paper in Nature magazine in 1953 showing that the most common form of DNA that we see today was known as the B form of DNA, and they figured that out. They didn't figure it out completely on their own, and that determination was made possible largely because of the fact that they stole data, and they later literally admitted it, from a phenomenal young female biophysicist named Rosalind Franklin. I'll tell you briefly the story of this because I think it's one that students really ought to hear. There's an excellent book called In Search of the Double Helix by James Watson, where he actually tells you everything that I'm telling you here. If you want a good book that's easy to read about how science sometimes proceeds, I recommend this book to you. Okay? Well, how did this happen? Well, it turns out that in the late 1940s and early 1950s, it was quite clear by that point that DNA was the genetic material. As late as the 30s, people thought that the genetic material might be proteins. They didn't know. But there were definitive experiments that showed that DNA was the information that was being passed from cell to cell. Okay? So by that point, the race to determine the structure of DNA was on. The race was partly stimulated by the knowledge determined by a young scientist who got his Bachelor's of Science degree from Oregon State University, and that was Linus Pauling. Linus Pauling discovered two very important structures in proteins, alpha helices and beta strands. We can credit Linus Pauling with that. And the knowledge of those sort of helical arrangements that were in DNA got people thinking about, well, what might, in proteins, we got people thinking about what might be in DNA. So there was a race, and there literally was a race to find out who could determine the structure first. Linus Pauling was in it, Watson and Crick were in it, and Rosalind Franklin was in it. And she was having to do a lot of it essentially on her own. She was a postdoc working in a laboratory in England. She was the only person on the face of the earth who was able to determine crystallographic information, or X-ray diffraction, I shouldn't say crystallographic, but X-ray diffraction information from DNA. Nobody else was able to do this. And she had information on her, or she had data from her analyses that actually showed to anybody who knew X-ray diffraction that this molecule was a helix and the spacing of the helix, how far apart the bases were. Okay? Well, Watson and Crick learned this, and they got this information from her mentor, the person that she worked for in the laboratory in England. They went to a big meeting on DNA structure, which was being held in England at that time, and her mentor was there. He had her data, and he said, "You want to see it?" So he shows it to them. That's how they see it. They didn't actually go and steal it, but he showed it to them without her permission. Okay? Linus Pauling was also in the race to determine this structure as well. Linus Pauling was a scientist in America, and Linus Pauling was, at that time in the early 1950s, very active in what was a very simple peace movement. The peace movement was to restrict the testing of nuclear weapons in the atmosphere. We used to test nuclear weapons in the atmosphere. We'd blow up bombs in the atmosphere, and the radioactive material would blow all over the face of the Earth. It's a true story. Linus Pauling said this is ridiculous. So he organized an effort to stop this from happening. Okay? As a result of that, he was known as a peacenik, and he was, in fact, restricted from leaving the U.S. to go to this meeting. It's a true story. It took a visa to go to England in those days, and the U.S. State Department says, no, you can't go. Linus Pauling didn't get to go to that meeting. Linus Pauling didn't get to see the data that Watson and Crick got to see. Now, whether he would have looked at it and done something with it, I don't know. But I think how unfair life is. Rosalind Franklin has the data. Okay? She doesn't get to do it. Linus Pauling wants to go, doesn't get to go because he's a peacenik. The two people who were able to go saw it and determined the structure of DNA. History is an interesting thing, isn't it? History might change as a result of things that we see. So, the sort of byproduct of that was, of course, Watson and Crick were given the Nobel Prize in 1958, I believe, for their knowledge of the structure of DNA that came from that, and a very well-received recognition. They did contribute to it as well, so I don't want to say that they did nothing in this process. They had some very important insights into doing this, but Franklin's data was a key piece. Rosalind Franklin did not win the Nobel Prize, and here it was for a different reason that was very unfair. She died. She died in 1957 as a result of a very rare type of, I believe, ovarian cancer. And it is thought today that she got that ovarian cancer as a result of the fact that when they did X-ray diffraction in those days, they did it without shielding. So, sad, important thing. But it's an important story, and I want everybody to know that story because I think it's worth telling and repeating. Anyway, getting back to actual biochemistry. The B form of DNA was the form of DNA that Watson and Crick discovered. There are other forms of DNA. And it turns out that in the very same issue of Nature, Rosalind Franklin actually reported the structure of the A form of DNA. Okay? Well, why is it that Watson and Crick are famous and Rosalind Franklin, at least for many years, was almost an unknown? And the reason is because the B form is, we know today, the most common form that we find in solution. And it was pretty much felt that was going to be the case when Franklin discovered the A form of DNA, because the A form of DNA could only be, it would only occur when she took the DNA and she dried it way down. Okay, well, cells are full of water, and so the thinking was, if this only happens when it's very, very dry, it's probably an artifact. It's not some real form of DNA that's important for cells, whereas the B form, which doesn't require this strong drying down, probably is the major form. And the second part was correct. The B form does form in cells. We know today that the A form also occurs in cells. Okay? The A form of DNA occurs in cells based on sequence specificity. That is, the sequence of bases. There are some sequence of bases that will favor the formation of A form of DNA, and many sequences that will favor the formation of the B form of DNA. Okay? What the A form is more notable for is the fact that double-stranded RNA, or RNA-DNA duplexes, that is where RNA is paired to DNA, those are always in the A form. So it turned out not to be an artifact. It turned out to have some significance. One of the things about the A form of DNA is that it appears to make the DNA bend a bit. And that may not be a trivial thing. Because if we think about the length of DNA that exists inside of cells, okay, you have enough DNA in every cell of your body to stretch out 7 feet. One cell that's smaller than your eye can see holds 7 feet of DNA. It's going to take some bending. It's going to take some arrangement. It's going to take some wrapping to make that DNA fit in the confines of a cell. And the A form of DNA may play an important role in that process. Now, they are fundamentally different forms. I'll point out a couple things to you. They have what we call a major groove and a minor groove. The major groove is this big sort of gap here. Okay? The minor groove is a little harder to see, but it's a little gap right here. So if we were to go up the strand of a DNA, we would see a major groove, minor groove, major groove, minor groove, alternating up the strand of DNA. And they arise from the arrangement of the structures of the nucleotides comprising the DNA. The DNA is arranged so that the phosphates are on the outside, the bases are on the inside, and the phosphates link together the deoxyriboses on the outside. So on the outside, we have deoxyribose, phosphate, deoxyribose, phosphate, deoxyribose, phosphate. To each of these deoxyriboses, we have attached a base, and the base is on the inside. The same is true of the A form of DNA, and the same is true of the third form I'm going to show you, which is the Z form of DNA. But these differ from the B form in how they arrange things. You can look at this guy here and see, whoa, this guy is really smashed. It's hard to see the major groove and the minor groove. They're not nearly so pronounced. Okay? But they still have the arrangement of phosphates and sugars on the outside and bases on the inside. The A form uses A pairing with T and G pairing with C, just like the B form of DNA does as well. Okay. Now, there's a better depiction of the major groove. And again, this is the major groove. This is a minor groove that you can see right there. The major groove and minor groove are important, not just structural considerations, but they're important because these are places where proteins bind to DNA. So later we'll talk about transcription and proteins playing a role in turning on RNA synthesis and turning it off. Those proteins work by sequence specificity. That is, they recognize specific sequence of bases, and they actually read these in the major and minor grooves. They bind to these grooves, and they can actually look inside the DNA and determine the structure of the bases within there. Not surprisingly, they bind by hydrogen bonding. Okay? So hydrogen bonds, as we've seen in proteins, are very important. Hydrogen bonds hold together the bases, and hydrogen bonds are what the proteins use to read those bases as well. So where would we be without hydrogen bonds? We don't have anything that is as important as those guys are. This, we don't need to worry about the major groove side, et cetera, but this is trying to show you that proteins can actually look down, as it were, in that groove and see these bases. The reason I show you this figure is not to tell you that, but to show you, of course, something that you probably know, and that is, of course, that A pairs with T, and it uses 2 hydrogen bonds to hold together. G pairs with C, and there are 3 hydrogen bonds that hold that together. Okay? That is important, as we will see later, because that means that GC base pairs are stronger. There's more energy holding them together. If we want to pull them apart, it takes more energy to pull them apart. AT base pairs only have 2 hydrogen bonds, and we can pull them apart much more easily. A third form of DNA was discovered by Dr. Alex Rich at MIT back in the late 70s, early 80s. And at first, when it was discovered, again, it was thought artifact, something odd, not really relevant for cells. The Z form of DNA differs from the A form of DNA. It's a little hard to look at this and tell in the way that the strands twist upon each other. Okay? The way that the strands twist upon each other. This table, which you're not going to memorize, summarizes what's going on. The A form and the B form of DNA twist the strands together in a particular way that we refer to as being right-handed. It turns out that when you have 2 strands that are intertwined, there's 2 ways that they can wrap around each other. One is like I sort of draw like this, and the other is backwards to that. Okay? They're called right-handed or left-handed. The B form of DNA and the A form of DNA both are right-handed helices. And if you would like to know how to determine if something is right or left-handed, come to my office. I'll show you with a phone cord how you can determine if something is right or left-handed. You're not going to have to do it for the exam, so just do it if you're curious or whatever. You're welcome to come by, and I'll be happy to show you. But suffice it to say that the A form, B form DNA are right-handed. The left-handed form is left-handed. The strands are twisted in the opposite direction. And so it was thought when it was discovered that it was an artifact. It'd be very unlikely to have strands twisting one way and then all of a sudden twisting another way and then going back to the original way. It just seemed very odd. The Z form of DNA got its name from the fact that it really looks like a zigzag structure. It looks very different. It's hard to tell from that figure I have, but it looks very different from the A and the B forms. Okay? Look at the number of the dimensions here. So we've got, for example, the number of the rise per base pair. This basically tells us how far apart individual base pairs are. We see the base pairs are very stretched out, 3.8 angstroms away from each other compared to 2.3. No, don't worry about the numbers. But Z we can think of as being pulled up this way, A being very compressed, and B being somewhere in between. Okay? And this is also shown, it's a little hard to see from this, but we can see here in terms of the number of base pairs per turn of the helix, these numbers reflecting the fact that there's some very big differences structurally between these. Now, zDNA, as I said, was thought to be an artifact. But I'll tell you one other brief story, and then I'll get off the stories and we'll get back to more data. And this actually relates to an experience I had working with an undergrad in my HHMI program back about 10 years ago. About 10 years ago, Dr. Xing Ho, who was at that time a professor in my department, had been a postdoc with Alex Rich back in MIT, the lab where they discovered the structure of zDNA. So Xing was very interested in the structure of zDNA. And like the A form of DNA, he was interested in what sequences of bases favored the formation of Z. So he did a lot of X-ray crystallographic work and was able to determine what sequences favored the formation of Z. By the time Xing was at our lab, it was known that Z did form in cells. So he wanted to know where, if I have a stretch of DNA, where does Z occur? So he had the idea for a computer program, and he gave it to this student in his laboratory and said, here, you want to have a summer project, why don't you write a computer program that uses this information, and it can predict where in a stretch of DNA a Z will form. You just look at the whole sequence of DNA, and in that sequence, by using that sequence information, you can say, okay, this region here is in Z, this region is in B, this region is in A, etc., mapping an entire chromosome. Well, I say an entire chromosome because it turned out that that summer, while he was working on his computer program, the complete sequence of the first human chromosome, chromosome 22, was published. Okay? So the human genome wasn't done yet, but the first chromosomal sequence was published. And so he thought, oh, wow, this would be cool. Let's analyze human chromosome 22. So they did. This undergraduate was a sophomore. This sophomore became the first person on the face of the earth to know and completely characterize a human chromosome. Okay? Now, he did it in a summer project. And I tell this story so that you guys know the importance of doing undergraduate research and getting out and doing things. He did it because he sought this opportunity. Now, there's an even better component to this story than that. Right? He looks at where the regions in this human chromosome where zDNA form, and he knew exactly the places where zDNA would form. And then the first question you ask after you know that is, I wonder if there's any significance to where they form. And one of the things they found when they analyzed it was that zDNA appeared to form very close to where gene sequences were. Now this was very important because when we look at a human chromosome, there are a lot of genes, but there's a lot of empty spaces between them. Identifying where the genes are is a bit of a mystery. How does the cell know where to do stuff? Well, if a cell has a little flag standing up there saying, here's zDNA, that may be very easy for the cell to recognize, oh, there's a gene here. Okay? So his work, he was the first author on that publication, and it was published in a scientific journal, and needless to say, is a very, very well-cited reference. So a very cool set of experience. So anyway, zDNA is important. We don't fully know all the reasons that zDNA is there, but we do know that it does form, and that it forms, at least in some cases, in the regions where we have genes. Okay, well, there are a lot of variables in DNA structure. One of the variables in DNA structure is the fact we think about the strands twisted around each other. The strands twist around each other in one way or the other. And we can imagine if we took a rubber band and we started twisting up that rubber band, what's going to happen to the rubber band? Well, it gets all coiled up, right? It doesn't look like a flat rubber band laying out here on the table. It gets all coiled up. It gets very small. And it turns out that the strands in DNA can be twisted in the same way. Right? What I've just described to you is something called supercoiling. Okay? Supercoiling. What supercoiling does is it causes DNA to really be strained, as it were. The strain in DNA arises from how many twists and turns we put into that DNA molecule. Okay? What Watson and Crick discovered was that DNA has, the B form of DNA, which is the most common form of DNA, has about 10.5 base pairs per turn. Meaning that if I go one complete turn of the DNA, I've gone 10.5 base pairs. It turns out that that amount of base pairs per turn leaves DNA in what we call a fairly relaxed state. No tension. It's happy to sit there and do anything else. If I start twisting it up more, or if I start twisting it less, the DNA will be under strain. Okay? So only when it's 10.5 base pairs per turn is it relaxed. And that sort of makes sense. Think of the rubber band. If I twist it one way, it gets coiled up. If I twist it the other way, it still gets coiled up. Right? Okay. Well, that brings to mind, brings up the topic of topology. So we have to spend a minute talking about topology of a nucleic acid, specifically in this case, DNA. The figure in your book is okay. It starts out by showing us a long stretch of DNA that has 25 times the strands cross each other. If we had 10.5 base pairs per turn, okay, let's keep it simple. Let's say we've got 10 base pairs per turn to make it relaxed, okay? We would have 250 bases here, right? So if we had 10 base pairs per turn, we'd have 250 bases here, and everything is fine and dandy. If I increase the number of turns, what happens to the number of base pairs per turn? Well, it's going to go down, right? The denominator will get larger, right? So the base pairs per turn will get smaller, and that DNA will be under stress. What if I unwind it? Okay. Well, it's also going to be under stress, and I'll have more base pairs per turn. If I took that original molecule and I closed it in a circle like I see here, I would have 25 turns, okay, going around this circle. We can count them, etc. This guy's relaxed, meaning it lays here flat on this screen, or it lay here flat on the table if I had it laying out like that. It's not going to get all kinky like the rubber band is going to get if I muck with it. What if I start with that original guy, and instead of having 25, I decide, oh, well, let's unwind it a couple times. So now I've only got 23 times that the strands cross each other. I have the same number of base pairs, so the number of base pairs per turn has just gone up, right? I have unwound this complex. What happens when I do that? Well, what happens when I do that is one of two things. This shows that original circle with 23 times of crossing over each other, and then nothing left for these guys to do. These guys here, of course, the strands want a base pair. They want to do their thing. This guy right here is stressed. Okay? You think you're stressed around an exam? These strands are stressed. Okay? Now, when a molecule is stressed, it responds to that stress to try to relieve the stress. And that's shown here. What happens is this guy really, really, really wants to have 25 base pairs per turn. Okay? I'm sorry. He wants to have 10 base pairs per turn. Wants to have 25 turns, right? Wants to have 10 turns. It wants to have 25 turns, 10 base pairs per turn. I can't get that out of my head. Okay? So it rearranges itself. And it rearranges itself just like that rubber band does by now starting to kink up. Well, that kinking up allows 2 more turns to be built into the molecule. Now let me go back and show you the previous figure. Notice LK, TW, and R and WR. In the strain molecule, we had 23 times it crossed. These are turns. Okay? And the number of times the strands cross each other. All right? Back up. I'm going to confuse you. Let me not start with turns. Start with linking number. Okay? So the linking number is what I've been calling the times it crosses each other. Okay? 23 times the strands cross each other. The turns are also the number of times the strands cross each other, but also the number of times the duplex crosses itself. Okay? The duplex is not crossing itself here. The duplex has not crossed itself at all. However, because this guy is under stress, the duplex goes back and crosses itself. And that is here. Okay? Now, there we are. This guy has now crossed itself. It's crossed itself. It's uncrossed itself here. Okay? So that we have 25 crossings over that have happened in this thing where we had 23 before. The number of base pairs per T is back up to 10. Before we had 250 divided by 23. Now we've got 250 divided by 25. We're back to 10 base pairs per T. Does that make sense? No. Let me start over. Let's start over. This is what I'm describing sounds very complicated, and I'm probably making it worse than it is. So let's start here. I've got a duplex. This duplex has, in this case, 250 base pairs, 10 base pairs per turn, and it's relaxed. It's laying there on the table. It's not doing anything. Okay? We have 3 numbers that are of importance to us with respect to this, the topology of this molecule. Linking number, twisting number, and writhing number, LTW. Okay? There's an equation that describes them. The linking number is equal to the twists plus the writhes. Now, it's the twists that are relevant for the base pairs per turn. Okay? When I talk about base pairs per turn, I'm talking about that T number. Okay? Cells want to have the appropriate T number, not cells, DNAs want to have the appropriate T number that allows them to be relaxed. And they will strive to get that. In this case, this guy is already relaxed. So when I close the circle, what do I see? Well, when I close the circle, I see I've got 25 turns. I've got 25, this is called linking number, and I'll show you that in a second. Okay? And it has 0 writhe. Now, writhe is the number of times the strands cross each other. That is, the double strands cross each other. This guy hasn't wrinkled up yet. There's no double strands crossing each other. So the linking number 25 is equal to the twists plus the writhe. 25 equals 25 plus 0. On the other hand, if I take and I untwist this guy, okay, so I take 2 twists out of there, what do I see? I see that I now have gone to 23. This guy is under tension, but in this case it still hasn't rearranged itself, so it hasn't writhed yet, which means therefore that the linking number 23 must be the same as the twist plus the writhe. The linking number has got to be 23. All right. Now, this guy, because it's under tension, rearranges itself. And now what it's done is it's put 2 crosses of double strands over double strands. In this case, the writhe is -2. If it twisted the other way, it would be +2. Well, how do I know which one it's going to be? You know which one it's going to be based on the equation that we have. Okay? The equation that we have tells us that that linking number, we're not going to change. The cell wants to get the proper number of twists so that it has the right number of base pairs per turn. So it twists 2 more times. In order to twist 2 more times, it's got to writhe in the opposite direction 2 more times. So that linking number equals twists plus writhe. Okay? So linking number, once we've closed the circle, does not change. Linking number is fixed at that point. Once we have done that, the circle is closed. We're stuck there. Now you might say, well, when do we do this in the cell? It turns out that long DNAs, like we have in chromosomes, behave as if they're in circles over long stretches. They behave like they're in circles. That is, that they can't adjust their linking number. All they can do is adjust their twisting and their writhing. So long DNAs, like we have in our chromosomes, behave very much like this. Now, I'm not going to do the calculations further, but if you have questions about calculations, please come and see me. There's a couple problems in the book I've assigned, and I'd be more than happy to meet with you about this. But I want to show you one more thing about this. Notice this rearrangement right here. This guy was under stress, right? And when it was under stress, it could either do the writhing thing that I showed you, or it could sit there and exist like this, a looped out region. That looped out region, we will see later, is important consideration for transcription, and in some cases for DNA replication as well. All right. So the topology of these guys is very important for us to consider. And the topology, and here's an electron micrograph showing the same sort of thing that you've just been seeing. This is a real live picture from an electron microscope of 2 different DNAs, one of which is relaxed, and one of which is what we call supercoiled. It has a positive or negative writhe. All right? Notice the difference in size. Bacterial cells are very small. They're very tiny. In order for them to fit their circular chromosomes, and by the way, this is a bacterial chromosome. In order for them to fit their circular chromosomes into their tiny confines, they actually supercoil them so that they maintain a smaller size, a smaller space. Okay? Well, how do we get from this to this? How do we get a DNA to become supercoiled? We don't have little fingers that come in there and unwind it and then put it back together. It turns out cells have enzymes called topoisomerases that do just that. Okay? A topoisomerase is an enzyme that will change the writhing of a DNA. It will change the writing number of a DNA. That change can be in a positive sense. That change can be in a negative sense. Most commonly in cells, cells have a negative writhe. They're underwound. But positive and negative both have the same effect. They both wrinkle up no matter what you do. Okay. Well, this simply shows a topoisomerase bound to a DNA. And the mechanism is not really essential for our purpose, so I'm not going to go through that. But let me just describe what these enzymes are doing. There are two major classes of topoisomerases that we will consider here. They're called type 1 and type 2. Type 1 topoisomerases in E. coli, okay, have an interesting mechanism. All right? Let's imagine I've got this DNA and I've got it just sitting here, let's say, very tightly wound. Okay? So it's really scrunched up. It's got a lot of tension sitting right there. We'll find situations during DNA replication where tension can be created as a result of the replication process. Well, if tension builds too much, that DNA is going to form a knot. It's going to get so tangled, it's going to get so wrinkled that it won't be useful for anything. So cells have topoisomerases to relieve that tension. That's part of what they do. All right? So I've got this DNA. It's very tightly wound up. Let's say it's in a circle, so it's easy for us to imagine. And there's no way to let it loose. It can lay there wrinkled up. Or the cell can say, well, I need to do something here. Let's relieve that tension so it doesn't sit here in a knot. What a type 1 topoisomerase does is it binds to the DNA, okay? And it does something we call nicking the DNA. That is, it breaks one phosphodiester bond in one strand. The other strand is left alone. Okay, everybody with me? So it nicks one strand and leaves the other strand alone. Now, if that guy is really wound tight, what's going to happen when you unwind that one strand, when you break the bond in that one strand? That strand is now free to move. It's going to go zzz. Relieve that tension, right? Relieving that tension. So type 1 topoisomerases are very easy to understand in terms of their mechanism of action. They allow that thing to relieve its tension, and then they simply recreate the phosphodiester bond, and now it's relaxed again. Make sense? So type 1 topoisomerases nick one strand and make this happen. Type 2 topoisomerases have a specific name in E. coli. They're called DNA gyrase, G-Y-R-A-S-E. And they have a mechanism that's a little harder to understand. In simple terms, instead of nicking one strand, they nick two strands. Now you may not think about it at first, but that actually turns out to be topologically a lot more difficult for us to consider. Because in the one case, when we nicked the one strand, the other strand was holding everything together. This first strand just wound around it, and then finally it came back to rest. When I nick both, now there's nothing holding everybody together. Okay? This guy could go flying apart. Well, you don't want it flying apart. You don't want your DNA ends to go flying apart. So this topoisomerase, this type 2 topoisomerase, has to manage to keep the ends together while this swiveling all is going on. Okay? The mechanism is quite a bit different than the type 1 topoisomerase, so I'm not going to go through the mechanism of it. But I want you to understand the problem of nicking both of those strands and still holding everything together. As I said, these enzymes can make positive or negative changes in writhe. Okay? They can actually create superhelical tension. They don't just relieve it. They can create it as well. If you're going to create it, it requires energy. DNA gyrase requires ATP. The type 1 topoisomerase I described to you does not. Okay? So making these topoisomeric forms requires energy in many cases, and topoisomerase 2, that is DNA gyrase, requires ATP in order to do that. Okay. And here is the problem. Let's just blah, blah. Okay. Let's see. We've got 1 minute for a song. What do you say? Yes. Okay. Quick song. It's a short song. It's to the tune of an old Simon and Garfunkel tune, which you guys probably never even heard of, called Feelin' Groovy. It's called Major Groovy. It goes, the DNA forms A and B. A base is complementary. Despite the similarities, they differ in their major groovies. Na 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 na. Major groovies. Transcription factors with their binding, cause DNA to start unwinding, holding it aggressively by forming bonds in major groovies. Na 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 na. Major groovies. For proteins, the key to sequence ID is hydrogen bonding, each base pair unique. Purine, pyrimidine patterns discrete in DNA's most major groovy. Na 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 na. Major groovy. Okay, good enough. See you guys later. Yes, ma'am.
Are you taking the test in here, in your office, in a different room?
You'll come to my office, and I'll take you to another room there.
Okay.
Okay.
And I actually looked up Bubble Boy, because I've been watching last year's lectures and this year's.
Yeah.
And I got so curious, and his parents had actually had a child before that died of it, and the doctors encouraged them to try another and do a marrow transplant, but his sister wasn't a match. And then they tried to do unmatched anyway, and she apparently had...
Yeah. Sad story.
Something. Epstein-Barr. That's what
Epstein-Barr, yeah.
Epstein-Barr that didn't show up.
Yeah. Yes, ma'am.
So we were talking, I'm in microgenetics,
Yes.
so we already went over Topoisomerases. Are they found in eukaryotic cells or just...
Every cell on the face of the earth needs them. Because as we'll see in DNA replication, that the replication process creates tension.
all that tension,
and
right.
so you got to deal with it.
So are they the same mechanism, the same type of enzymes, or are they called Topoisomerases?
They are called type 1 and type 2 based on whether they cut one strand or two ends.
And so they're the same in eukaryotic?
The same general mechanism, yes.
Okay.
And there's some that are unique to E. coli. I'll mention one of them maybe next time. It's kind of interesting about its role in replication.
You made my day by picking Feeling Groovy.
You like that?
Do you know Natalie?
Natalie, I know a lot of
Natalie
Natalie's.
Rich. She's in this class.
Okay, yeah, Natalie, yeah.
She hates that song with a fiery passion, but we're roommates, so I play it all the time, really loud. It's
Well,
a joke.
now you have a recording of me doing it, right?
I know,
So, what's up?
Kevin, does topoisomerases requires energy
Yes.
to uncoil or coil or?
It requires ATP energy in its mechanism overall. And that's partly because it has to break and remake phosphodiester bonds on both strands.
Okay.
And that energetically takes some energy to do.
Okay.
That's why.
And it can